The last macromolecule we're going to talk about are nucleic acids. So nucleic acids are the polymer, and the subunit or building block they're made of are nucleotides. So that's the monomer. There's two different types of nucleic acids. We have DNA and RNA. So how they got their name, DNA is deoxyribonucleic acid, so you can see where the DNA came from. And then RNA is ribonucleic acid, so you can see where the RNA came from, which is their abbreviation. Okay, so the functions of DNA and RNA. DNA is going to store the genetic information, and then the DNA contains genes, and the genes are going to code for proteins. And then the RNA is actually going to help make the proteins. And then this is just a picture of DNA, and you can see it's a double helix there. Okay, so when we express the gene that's in the DNA, we're going to be making a protein. So genes code for proteins. So it's actually a two-step process. So here we are, like maybe in one of our cells. So here's the nucleus. So our DNA is stored in the nucleus of our cells. So the first step is called transcription. So transcribe just means to copy. So we're just going to copy the DNA, and the copy of the DNA that we make of the gene is called messenger RNA or mRNA. And then we're going to send the mRNA out of the nucleus out into the cytoplasm, where we've got some protein making machines called ribosomes. And then the ribosomes are going to do our next step called translation. And translation, we're just going to have the ribosomes read the code that's on the messenger RNA, and they're just going to figure out what amino acid to put in what order to make the protein. And so that's how we go from the DNA to make the protein that the gene is coding for. Okay, so let's take a look at the nucleotide, the monomer, that we're going to join together to make our nucleic acid. So our nucleotide is made up of three parts. We've got a nitrogenous base, and these nitrogenous bases, there's 5 different ones. So these are the A's, C's, G's, T's, and U's that we'll be talking about more later. And then we've got a 5-carbon sugar. And in DNA, the sugar is deoxyribose, and then in RNA, it's ribose. And that's why there's a D in DNA and an R in RNA. And then we've also got some phosphate groups. Okay, so here's just the deoxyribose in DNA, and then the ribose in RNA. And the only difference, if you're interested, you don't need to know this, but you can just see that there's an O missing in oxygen in the deoxyribose, where there's an oxygen here in the ribose, because D just means it's removed. You've removed that oxygen. So deoxy just means that oxygen has been removed from that ribose sugar. Okay, so the next thing we're going to be taking a look at is just the differences between DNA and RNA. So DNA is double-stranded, so we've got 2 strands, whereas RNA is single-stranded, so just 1 strand. And then we talked about the difference in the 2 sugars, so we've got deoxyribose in DNA and ribose in RNA. So if you just remember, D goes with DNA and R goes with RNA. That's an easy way to remember that. And then with the letters that are used in DNA, we're going to use G's, A's, C's, and T's. And then in RNA, we're going to use the same letters, G's, A's, C's, but instead of a T, we're going to use a U. Okay, so there's T's in DNA, but in RNA, we've got U's instead. So RNA is going to use the U's, but DNA will use T's instead. So that's a big difference there. Okay, and then just some more pictures. So DNA, you can see, has two strands, double-stranded. And you can see how we match up the A's and T's and C's and G's. And we're going to be learning about this more later, but I always remember apple tree and chewing gum. That's how I remember which ones go together. And then you can see that RNA is just a single strand. Okay, so we've got all these nucleotides joined together, and we've got a single strand of the nucleotides to make up our nucleic acid for RNA, and then for DNA we've got two strands. Okay, and then just a review for our macromolecules here. So remember that for the carbohydrates, the monomer would be monosaccharides and the polymer would be polysaccharides. For proteins, the monomer would be amino acids, and we join those together and make our polymer, our proteins. And then for nucleic acids, the monomer will be the nucleotides, and then we join those together to make our polymer, the nucleic acid, which can either be DNA or RNA. And then for the lipids, remember that they don't have one particular monomer because it's such a varied group. And so there's really no polymer for the lipids. So they are a macromolecule, but there's really not a polymer for the lipids because they're such a varied group. We have three different types of lipids we talked about. We talked about the fats or triglycerides. We talked about the phospholipids, which make up the plasma membrane or cell membrane. And then we also talked about the steroids, which include cholesterol and our steroid hormones. And these we just grouped together because they're all hydrophobic. None of them like to mix with water, whereas all these other ones, the carbohydrates, proteins, nucleic acids, those are all hydrophilic, so they like to mix with water.